Our next speaker is Dr. Victoria Roberts. She is an associate project scientist at the San Diego Supercomputer Center, and her lab is engaged in studies of protein-protein interactions and protein-DNA interactions. And today she's going to tell us about some new things she discovered relating to the structure of Hiv integrase when bound to DNA. And her title is C-terminal domain dimer organizes the Hiv integrase assembly.
Thank you very much for the opportunity to present this work. We're going to go to a smaller scale. We're going to go to sort of the atomic scale. I'm going to talk about Hiv integrase, one of the three enzymes that are available for the HIV, for the retrovirus. And I'm going to focus on mainly the C-terminal domain. And funding for this project was available both from CFAR and from the California HIV/AIDS Research Program. So HIV integrase, its job is to insert the viral DNA into the host DNA. And the way it does this is it first trims both two nucleotides off the viral DNA ends, and then it inserts those into the host DNA. The first structure, partial structures of HIV integrase became available in the mid-90s, but none of them contained bound DNA, and the viral DNA is known for organizing the biological complex. What they did find out was that the catalytic domain that actually holds the viral DNA ends is a dimer, but that the two active sites of the dimer are not the right distance apart to insert both viral DNA ends. So it became clear that you needed two dimers to make the biological assembly. More than 15 years after the first structure of HIV integrase, the very first structure of any integrase, full-length structure with bound DNA, occurred. So it took that long, and there was a mammoth amount of interest in this structure. And that is of the prototype foamy virus in integrase, which I'm going to have to say PFV for. And as you can tell, the structure is rather complex. This really is a breakthrough structure. It's groundbreaking, but it showed how two dimers come together to form the complex. And what I'm showing here is structure without the DNA showing, and the structure on the, I'm confused, that would be on your right, is the structure with the DNA. And only two of the four chains are shown, and this is a little complex. So for a given chain, it starts out with the N-terminal domain, or the NTD, and that is linked by a long linker, shown in light blue, to the gray catalytic domain, which is connected by a very long linker, shown in orange, to the C-terminal domain, so that within a single chain, there's very little contact between the domains. This is shown, if you're not familiar, each bend represents the central atom of an amino acid. So at each bend, it represents there's an amino acid followed. It's an amino acid chain in a very simplified form. So this exciting structure really brings us to a question of, can it tell us about HIV integrase? And there's a few problems. One is that the linkers actually separate the C-terminal domain that are shown over here from the DNA. And cross-linking studies say there's interaction between the C-terminal domain and the bound DNA, viral DNA. The second problem is there's very low sequence homology between the prototype foamy-viral and the HIV integrase, except for the catalytic domain. And to give you an example of that, the linker that connects the catalytic domain to the C-terminal domain, shown in orange, is 50 amino acids in prototype foamy-virus, 21 amino acids in HIV integrase, and just 8 amino acids in a fairly closely related to HIV integrase from avian sarcoma virus. And so 8 amino acids is simply insufficient to span this distance. It just can't happen. And the 21 amino acids from HIV, the crystal structures, partial crystal structure, so a helix, it also cannot extend that distance. And finally, I'd like to say there is a hole between the two active sites. No protein is there, although we have to bring together 3 pieces of DNA. So before making a model, I thought, let's just explore the prototype foamy virus assembly. And I like to explore things by computational docking. Maybe we can find alternate configurations that might address some of the problems in the previous slide. So I'm going to take the individual domains and look at them and their interaction with viral DNA. And all of these components are shaped in the complex to interact with each other. So the rigid body docking I'm doing should reproduce those interactions unless a binding surface requires multiple components. If I only have one, I might not see the interaction. The program I use have developed in a lengthy collaboration between Lynn Tenike at San Diego Supercomputer Center and Michael Peake at the Scripps Research Institute. And the most important thing about it is it's a complete search over all space. It tests everywhere, and it calculates billions of configurations and gives an approximate energy between the two. And this program is particularly good at modeling electrostatic interactions between protein and DNA, which is very important for this project. So first, we just looked at a particular grouping from the structure of the catalytic domain. It's the N-terminal domain that interacts with it and the viral DNA. And they look like this in the crystal structure. So you just take the individual pieces and dock them. So if you take the catalytic core domain, the gray domain, and you dock the viral DNA to it, it's basically, does it match the crystal or not? And the answer is yes. And the way I tell that is within the top 30 ranked complexes, 22 are very close to the crystal structure. And with the total top 2,000, a very large number, also 250. So you just look at the top 30 and you go, that is the crystal structure. If you take the N-terminal domain by itself and dock viral DNA to it, you do not reproduce the crystallographic structure. If you take the N-terminal domain and combine it with the C-terminal domain, so you've got the gray and the blue, you get a beautiful fit. So basically, it seems like the interaction that starts it all is the catalytic domain with the viral DNA, and then the N-terminal domain fits in to complete the viral DNA binding surface. But when we take the C-terminal domain, here's an example of it in the crystal structure, and dock it to any one of these pieces, we do not match the crystal structure. So I thought the C-terminal domain contacts a whole bunch of things in the crystal structure, a little bit of the catalytic domains, both viral DNAs, and maybe the N-terminal domain. So let's make a construct of the 2 catalytic domains plus the viral DNA, and we're going to dock the C-terminal domain to that. And so we do that. We take this construct. First, we dock the N-terminal domain. Docks beautifully. 27 of the top 30 match the crystallographic structure, both positions. You'll show up both here and here. Do it with the C-terminal domain, no success. You add in all the domains. So you add the N-terminal domains to this, and you try and dock the C-terminal domain, no success. So that was very strange, but the amazing thing about it is that it's not that the C-terminal domain is just docking all over. It actually goes to a very specific spot. So compared with its position in the crystallographic structure, when you dock it, it goes to 2 symmetry-related positions that are centered right over the 2 active sites. And one thing that happens is the C-terminal domain is now closer to the catalytic domain, so the distance span decreases. And the other thing that happens is the C-terminal domain in this position now contacts parts of the viral DNA that were found by cross-linking studies. So the amazing thing about this is how strong a hit this is. 28 or 29 of the top 30 are in these two symmetry-related clusters. Within the top 100, over 3/4 are in this cluster. It's better results than I got by docking the pieces into their crystallographic position. It's kind of stunning and unnerving. So it lies right in the middle of the active sites, and it has a shorter distance that can now be spanned by the different lengths of the L2 linker. The problems with the docked position is potential clashes with both of the linkers in the original prototype foamy-virus structure that literally stretch right over this region. The orange linker that goes from the C-terminal domain to the catalytic domain completely moved out of the active site, no problem. The other linker, actually you can invoke an N-terminal domain from a different protein chain and move it out of the way also. But one of the key problems is the two loops of these guys overlap. They're filling the same space. So to solve that problem, I rebuilt that particular loop shown in green. And I looked at the HIV integrase C-terminal domain, and if you actually position it in the docked position, you find out that this loop actually is in great position to not interact with the other docked C-terminal domains. So I rebuilt the prototype foamy-virus loop to go in the same direction as the HIV. And the result is, so this is a view, now we're looking from the view of the host DNA. We're looking down into the active site. And those two loops that get rebuilt in either as they are in HIV integrase, if it's in the superposed on the docked position of the C-terminal domain from prototype foamy-virus, or the rebuilt prototype foamy-virus C-terminal domain, the loops now poke right into that empty space between the two active sites. And what they do is they insert a cluster of positively charged amino acids between the two viral DNA-bound ends. And the host DNA is going to lie right down on that surface and contact those charged residues. So now we have a protein domain providing a stabilization for the coming together of three pieces of DNA. And that helps neutralize the large negative potential that's created by trying to bring so many pieces of DNA together. This is a model of the HIV integrase built based on that docked position. And it's basically showing that the catalytic domain in gray and back has the linker that winds around and then comes to the C-terminal domain. And so now basically that group of three from the catalytic domain, the linker, and the C-terminal domain are now winding around the viral DNA. And as well, that C-terminal domain is touching the other viral DNA. So what's happening is now there's a central hub created by the C-terminal domain in the center between the two active sites that helps bring together the two active sites and determine the spacing of the two active sites, which determines the distance that the two viral DNA ends between the insertion points into the host DNA. So the conclusions are that the prototype foamy-virus and HIV integrases, my computational experiments suggest that they have a similar position for the C-terminal domain, but it's not what's in the crystal structure. And that the proposed position accommodates the huge variation in the linker that connects the C-terminal domain and the catalytic domain. It's the main determinant of how the two active sites arrange, and a central hub, and that it helps neutralize the large amount of negative charge created by the close proximity of all the DNA. And what I'd like to say is that given that it's a computational study, it would be nice to have experimental evidence to try and support this. And so I'm also involved in collaborating with Cheng Li at UCSD in the mass spec lab and we're performing solution studies of HD exchange that's then analyzed by mass spec to see if we can find, identify changes in integrase in its different states. Thank you.
It's a nice study. We have time for a couple quick questions.
So it's a beautiful study and a very complicated system to address by molecular docking, and I just wanted to check that I got it right. The initial docking was rigid body, right? But
Yes.
then you had to introduce flexibility of the loop to actually reproduce the geometry that is consistent with the catalytic mechanism.
Yeah.
Is that right?
The docking is all rigid body, yeah. Yeah. Did that answer your question entirely?
Well, you had to remodel the loop.
Yes. Well, actually, the prototype foamy-virus, we used the loop, but we've done the same calculations using the HIV integrase partial structures. And yes, the first thing you have to do is you have to transplant the active site loop and the full catalytic site from the prototype foamy-virus into the HIV virus. And then it works really well. The N-terminal domain, the catalytic domain, and the viral DNA shaped as in prototype foamy-virus, but with the right DNA sequence come together very nicely.
Thank you.